and so it's going to talk about the same type of models, but at a more preliminary stage to understand multisensory integration of stimuli in the mouse. And so I'm going to present the different parts of the experiment and some of the calcium imaging setup that is there, and how we use decoding to analyze the data. All right. So let's start. So if you don't, if I'm not speaking loud enough, let me know. Feel free, as Damien said, to ask questions whenever you want. So it's meant to be didactic and maybe more dynamic. So I start with so the basic of making classification pipelines to extract markers from all brain MRI data. So typically when we get functional data in the form of ball signals that correspond to the activity over time here for different regions corresponding to parcellation, you want to use them to make the prediction of behavioral score or pathological states, to discriminate, for example, patients from healthy controls. And we would like to combine this information with structural MRI data that's concerned with the connectivity, so the white matter fibers that go between the same regions. And so a pure machine learning analysis would be to align these different types of data in a prediction pipeline, a classification pipeline. But what we did was to use a model, so to combine these two types of information in a dynamic model where you've got different regions that are connected in the brain. And I'm going to explain how this connection with weight are the effective connectivity, so that measure the strength of interactions between the different regions, is going to be estimated from the fMRI signals and constrained by the anatomical data. And this, in fact, so this effective connectivity here is going to be the signature, so the value that we're going to use for the prediction of our behavioral scores or pathological states. So the idea here is so to bring in the dynamic model the two types of data, and so in a sense, this in this dynamic network here, so the different regions are connected by some weights. So we've got directional interaction between the regions, and they explain how the signal, the fMRI signal, they propagate between the different regions. So that's we're going to fit this kind of model to the data, the fMRI data, and we're going to tune in this model only the connections that exist in the anatomy. So we are going to decide what is the substrate of connectivity, so the topology of this network, using the anatomical information. And so the point of this model is to capture the dynamical states there via this effective connectivity that is going to describe how subnetworks of region in the brain, they communicate, and this communication is going to depend on different conditions, or for pathologies or for cognitive tasks. So to go a bit in the details of the different types of connectivities that we're going to talk about here. So we're going to talk about structural, functional, effective connectivity. And so as you may know, so functional connectivity is a measure of the statistical dependencies between the activity of different brain regions. So for example, here as a sketch, we've got 4 different regions that exhibit some strong functional connectivity. So for example, some strong correlation or synchronization between regions A, B, and C. And so the point here is to understand from what you observe from the activity, where this strong synchronization comes from. And if you look now at the underlying connectivity, so we have got some information about what is the actual anatomical network that's in the brain. We can see that there is a connection between region A and B, B and C, but not a direct connection between region A and C. So the question now is how the strong correlation between A and C emerges from this network. And so the concept of effective connectivity is then to try to estimate this directional interaction. So meaning that we're going to take this template of anatomical connectivity and estimate weight for each direction. That is going to explain that in this case, it's region A that is driving region B, and region B and C are strongly coupled. And this as a result, so that's a mechanistic explanation why you observe the strong correlation between the three regions A, B, and C. So in that sense, that's how we bring together structural, so anatomical data and functional data together in the same model, with effective connectivity being like an explanation of how it works, right? Okay, so that was a bit of a conceptual explanation of the relationship between these connectivities of different types. And so the main point here is that to do this, we need to decide on a model of network dynamics that's going to explain how the different signal propagate from one region to another. So that means that this weight of effective connectivity to explain in the model who's affecting, which region is affecting what other region to which magnitude, let's say. And as a result, this model, when fitted to the data, describe via the effective connectivity like a mixture of the effect of synaptic strength, local excitability, neurodegeneration, and so on, that that explain the brain communication. So just for those that are familiar with the dynamic causal modeling by Stone that introduced this concept of effective connectivity. In the present model, there's no hemodynamics. So if you want to talk more about this later, I'm happy to go further, but I won't go in the details right now. So now we are going to have some data, and we want to fit the model to the data to estimate this connectivity weight, effective connectivity weight. Right? And so why does this work? So basically, when we look at the fMRI signals that are here, we need to decide for a model that is going to reproduce their spatio-temporal structure, right? And so when we look at the autocovariance of the Ball signals, which is represented here, so on the x-axis, you've got the different lag for the autocovariance, meaning that that's somehow a measure of the correlation from time to the next time point in the Ball signal. So if you're not familiar with the autocovariance, you can think of it as a kind of memory property of the signal, so how slowly or how fast it changes in its pattern. And so this kind of memory depth of the signal can be quantified with the time constant. And so when we look at this autocovariance, for the Ball signal, so we can see that all the regions, there's somehow the same profile of autocovariance, which is shown here in the plot with the log axis and with the time shift. So this is just half of this small inside here, but plotted with the log axis here. And the main point here is that somehow all these brain regions, they've got the same kind of memory base. So we can model them with a dynamic model, which is a network model, which is of this form. So don't be scared, that's going to be the only equation of the presentation. And long story short, this model, which is a dynamic network model involving noise, with some stochastic process, so to say, has different types of parameters. So parameter C here is the effective connectivity. This time constant is the one that I presented just before. We can estimate it from the data as well. And this input noise here, the DW, has another structure as well. So it's like white noise that is inserted in the network. And it has a given input covariance. And the point here to fit the model to the data is to estimate these different parameters from the signal. So, yeah.
How you are estimating the time constant then in the study report?
Yeah.
And how you are deciding on this TX?
Yeah. So one thing that is bit hidden here is that all these autocovariance profiles for the different. Okay. And so what is represented here in the scale is related to the slope of this exponential decay related to this time constant. Yeah, I was not sure I would go into the detail, but thanks for the question. And so because they all have an exponential decay, we just checked that they have the same time constant. And because of this, the Bold signal are well suited to be represented using this Einstein wave process that naturally generates a signal with exponential decaying covariance.
Is the same time constant, time constant for every
every region has the same time constant.
Okay.
In the model, it's possible to refine that, but because in the data we see the same time constant, then we can use the same for all of them. And that's a bit of, well, that's an important point numerically because this helps a lot to have a strong estimation of the weight. So that's one made in 2016, and there we compared different methods for estimating the connectivity because we want to do that at the level of each fMRI session, meaning that we don't have a lot of points to estimate the connectivity weight. And so the point of that study was to estimate robustly this EC weight as best as we could at the level of single fMRI session. And to do that, we needed some approximation or tricks, and this is one of them. All right. So this estimation is done using a gradient descent, and it's actually made using the functional connectivity of this Bold signal, but not only the functional connectivity like the usual correlation without time lag, but also the functional connectivity involving a time lag. So that means that we use a measure of the propagation of the Bold signal to estimate the directed connectivity of the weights here, right? And so why is that important? So I said that we're going to estimate all the parameters in the model, not only the noise, the input noise with that was the sigma here, but also not only the effective connectivity, sorry, but also the sigma, the noise, sorry. And here is just like one slide to show why it's important. So here in this toy example, we've got two regions that are coupled like symmetrically, but one is receiving more noise, like larger amplitude of the noise compared to the other. And if you look at the cross covariance between these two regions, you can see a peak that is around here. So you can see that it's a moderate peak, but it's still a peak, which is on one side. And so that means that apparently from the dynamics, from the observed activity, one region is leading the other. But you can see here that the connectivity is symmetric. So the apparent lag is just coming from the difference in the noise amplitude. If you look at the different configuration, now we've got the same amount of noise in the two, but one connection in one direction is stronger than the other. And the apparent covariance, cross covariance is the same. So to really make the difference between this, which is important when we do the estimation, we need to not only observe the cross covariance, but also the auto covariance. In other words, when we observe the activity of the whole network, then we can make the discriminate between these two configurations and say whether one region is strongly driving another one because of the effective connectivity, or whether what we observe is coming from different levels of noise in inputted in each region. All right. So that's important in terms of interpreting our results later. And here just another configuration. Yeah, well, I skip this one here. And so after doing all that and checking our method on like toy example and like more synthetic data, the point was to estimate the model from real data. And so here's one example of what we did. So here we wanted to estimate the effective connectivity from stroke patients to try to use it as a biomarker for prognosis, or clinical prognosis. So in this data that we got through the Human Brain Project from Matthieu Gilson, so the patients were admitted at a time where they had a stroke, and then they had repeated MRI scan, both structural and functional, at 3 weeks, 3 months, and 1 year, and also some cognitive tests to estimate their score. And so what we wanted to do was to use the fMRI scan at the acute phase in 3 weeks to predict the cognitive scores 1 year after. And so just to give you some ideas, we had like 100 patients. So it divided in 4 classes. So some are strokes concerning the gray matter, some the white matter, some subcortical, so different types of patients, and 25 controls. And so there we had 5 parcellation of the brain in 300 regions. And we used like 4,000 weights. So we have thousands of ways to estimate for each fMRI session, which is a lot. And then that gave us a rich marker to make the prediction. And the prediction was made like with a train test cross-validation, which is classical here, but just to say that we really check that our prediction generalized to new subjects, new patients that we don't use for the training, all right? And so this is the result that we had. So in blue, you've got the prediction for the real data over 50 train test split repetition. So these are the distribution of classification accuracy when you use the effective connectivity, and here what you do what the classification that you get when you use the functional connectivity. In orange, we have like the surrogate chance level that we obtain by permitting the label. And so long story short, you can see that both for FC and EC, the accuracy is above the chance level. But the good point here, I mean, it didn't work for all the prediction, but that worked for this one, which was a difficult one. You obtain a better classification accuracy with the median of the EC accuracy being at the level of the top quartile of the FC accuracy. So that means that we can use information about 1 year before to some extent predict how the patient they're going to recover in terms of cognitive deficits. Right. And so from that was a good result because that was one of the first kind with prognosis from MRI. So we went further and we looked because you use all the effective connectivity links to make the prediction. We went to look which one are informative for the prediction, right? And so what does that mean? That means that these links that are represented in this brain here, they correspond to links that change in patients that have large number of deficits or small number of deficits. So it's a proxy for the reconfiguration of the cortex, of the cortical communication that contributes somehow to the recovery of cognitive deficit. Right? And so, as you can see here, there are a lot of intermediate links here. And when we plot them in this kind of circular graph, where you have different subnetworks like the visual, the somatosensory, the auditory, the default mode network, or the dorsal attention network. So in this circle, these links that cross the circle correspond to interhemispheric connection. And so you can see these connections here. And we analyzed were close to the lesions that were related to the stroke, and we found that no, they were more distributed over the whole cortex, and they were involving like high-level cognitive network, like the default mode network or the dorsal attention network. So to recover the cognitive deficits, so that means that a large, a distributed subset of connection, especially reconnecting the two hemispheres, the one that's been affected by the stroke and the other, has to be engaged in order to successfully limit the number of deficits. So that's it for this part. If you've got some question, feel free. Otherwise, I'm going to move on. Or do we go this time?
So don't be shy on Zoom. If you want to ask questions, please, you can just switch on the microphone and ask.
Okay.
We can hear you.
Right.
I only have one question about before.
Just speak loud, Sami.
Yeah, I have this question about the effective connectivity. You are only seeing that it's related to monosynaptic pathways. Like you can have effective connectivity to something that is not connected. Why is that decision?
So what we want to measure with this effective connectivity is like the modulation of the efficacy of the white matter connection that connects different brain regions. So this is why we want to do that is because it's like functional connectivity. It's going to change across the condition, like doing different cognitive tasks. And what you want to understand is in the same way that you study the relationship between structure and function when you put the anatomical connectivity in a dynamic model like the virtual brain to study the pattern of activity and how the two relate together, like structure and functional relationship. What we want to explain here is how modulation of the anatomical pathways generates the pattern of activity that you see. So it's task-specific connectivity that is constrained by the anatomy because we don't want to say that two regions that are not connected, they interact. We want to explain their interaction via other third parties in the network.
Okay. So it's like a base or the ground is in the, in the structure. Okay.
It's one way to fuse the two information.
So Jaroslav has a question. So do you want to ask it?
Hi, thank you. I was wondering, in this informative EC links you have, they're mostly looking symmetric across both hemispheres. You mentioned that they're limited by the constrained by the anatomy. And here, is it correct that my impression that they map basically motor to motor or posterior visual back to visual circuits of the brain, and they actually rarely engage any kind of not cross-regional interactions, but actually across different types of regions, right? Like multisensory or motor to sensory feedbacks. And is it anyhow related to the place where the stroke has happened? That would be the second, but maybe somewhat related question.
Yeah, so about the second question, that's really what we tried to see when I mentioned this point, maybe too quickly, is that we really checked whether the informative links would be close to the region that we know from the anatomical disconnector, which was called like that. So whether these informative links were close to the lesions due to the stroke, the answer was no. And so the informative links that you see here, yeah, as you can see in this diagram, they often connect like mirror regions, like homotopic region, not the exact same one, but regions from the two hemispheres that are in the same functional subnetwork, so to say, like this resting state subnetwork. And one supposition that we made is that that may be a way to compensate by the same hemisphere the loss function in the lesion hemisphere. So that should be checked further, but that was the easiest explanation that we could.
Yeah, that actually, I agree. It hints a bit more like that maybe those brains of those patients have better homeostatic regulation, right? This is the pattern kind of broad and not specifically related to the place where the lesion or the stroke has happened.
Yeah,
right? And those people with better homeostatic regulation also happen to recover better.
Yeah, maybe one thing here that is important is that remember I mentioned that we had patients with different types of stroke, like subcortical, gray matter, white matter, and here in this study, in this classification, we pulled all groups together. So what you see is some pattern of prediction that is common across all these groups. And of course, the next step is to go within each group to see whether there are some specific pattern, maybe more related to the lesions for some of them or not. But at this level here, we are talking about some average effect across all types of stroke that we have.
Thank you.
I don't think there are other questions at the moment.
But I guess so the follow-up question of Yaro would be that maybe does this kind of reorganization is due to the way of living of the patient before, you see what I mean? Kind of the cognitive reserve kind of phenomenon, than depending on, you know, if you were active or whatever. I mean, I think that's what more or less what Yaro wanted to come. So
okay. That's one way. This reconfiguration across the sphere, yeah, it's one way to make use of the cognitive reserve when it's there. Actually, one thing that we also tried, but that was whether our new marker for fMRI was adding something compared to traditional markers for recovery that are age, education, and things like that. And the synergistic effect between the two was not strong enough so far. So maybe we need more data for that link, but we didn't see a clear effect there. But that's so maybe because they are a bit the same. They are a bit in line. Maybe what we see here is more strongly related to people who have this cognitive reserve that they can engage. That's a nice supposition.
So maybe I also have a question that you can.
Yes.
So effective cognitivity is directed
Yeah.
So, but then when you go to the important connections, so do you find the symmetric partners as well, or there are really directions? There's a left to right and not right to left that are more important than others.
So between the lesion and the same.
Yeah, no, the informative links. So, so if you find it in one direction, you also have the one in the other direction, which is informative as well. Or you really have specific directions which are informative and other directions which are not.
Yeah, we had
Very
asymmetry.
narrow the graph. So
Yeah, there are. Yes, but I can see whether there are double arrows.
Yeah, there are small arrows like here. So yeah, we found like asymmetry in the markers. So in this model. So here you don't have reciprocal connection in the marker, or you've got some of them, but
if you look at the cortical hierarchy, so it's more top down or bottom up that matter.
And what we found is something very distributed that involved both connection from the same hemisphere to the lesion one and the opposite.
Okay.
Not really reciprocal connections in the end, but something that was not specifically bottom up, top down. There was no obvious pattern there.
Okay.
From what we see. Once again, we only have 120 patients.
Only
30. It's a lot for this. But to go toward these statistics, you need a lot more, I think, to do that. And although we've got like good accuracy for the, I mean, way above chance, we are still not at the level of very good accuracy to have a pattern that would like generalize across so many patients. Yeah. So that's a limitation also of the data that are existing.
Okay, thanks. Second part.
Okay, so I'll just keep that saying that we also just quickly that we use also this same framework to study cognition and look at like a hierarchy for like a subject watching a movie, because Damian loves the good, the bad, and the ugly. But I had that slide. But basically, what we, in a very short description, what we are trying to do here is to look at communities of region at the whole brain level, but also something that we mentioned with Romain yesterday, which was like how reconfiguration at a more local scale could happen. And here you've got increase and decrease, so increase in brown and decrease in green between the connectivity of visual and superior temporal, so auditory systems, and how they converge towards the like fusiform or mediotemporal areas. And so this kind of diagonal here that is there, not fully, but still there somehow, is saying how this cortical hierarchy from auditory and from visual regions, sensory regions, is connecting to try to integrate the two modalities that are vision and audition in this passive task. So that's also something that we want to do. And just a note to sum up about the kind of model and how to use them with different, I mean, for markers. So in the model that I showed, like we've got this effective connectivity, which is a large set of parameters, thousands, that we use to for prediction. So depending on the model that you're going to use, you may have just a single parameter, like grouping from the models of Gustavo for the dynamic midfield, for example, that you're going to estimate from the data, or 1,000 as we showed here, and also for different models, some other things. But what I mean to say here is that this parameter that you estimate, they are your markers of the dynamic state of the brain. So there's a trade-off between having a lot of parameters for each biomarker and the robustness of the estimation, which of course, when you have so many parameters to estimate, you have a lot of noise. And so that's but that's the direction that we're trying to pursue, like to improve the realism, the richness of different biophysical model, and to try to represent them and to use them to not only for predictability, as I showed, but also to interpret what we showed before was how to use them for marker to understand the reconfiguration between the brain hemispheres for stroke. For the short example about cognition, it was to identify a hierarchy for multisensory integration. And so that's because the model is bridging the anatomical structure with the functional data that we can interpret it in terms of modulation of cortical pathways. So that's I think an advantage of the model. That's what I'm trying to push in the past year. And so last point is that it was still question whether the fMRI signal they have a temporal structure that is meaningful. And so this kind of result tend to say that yes, they can be used to make markers that are predictive. So I think it's also something to research here on more fundamental ground to relate the structure and function is how these bold signals that are so slow, they still can tell something about directionality between interaction in the brain regions. All right, so now let's go to the second part with multisensory integration, but now with the mouse and with colleagues from Jülich and Aachen, especially Damien Kuhn and Jonas Kolf from the work of Michel Scholl that developed the pipeline that I'm going to present, and Garion Lebefeld, Simon Mussel, who did the experiments also in the lab of Björn Kampa. So what we are going to look at now is an experiment where the mouse is going to have different types of stimulation, so visual or tactile. Tactile will be some air puff on the whiskers. And during the stimulation period, the mouse is going to receive from the two sides a number of visual or tactile stimulation that can be separated or go together. And the point of the task is to decide which side has received more stimulation. So it's a forced task. And to do that, the mouse is head fixed, as you can see here, which allows to record the brain activity using calcium imaging through a cranial window that you can see on the right. So here you can see the spots for the hairspurs, and there are also some spots for the rewards. This is the leaking spot for the reward, and this is one for the hairspurs. And so one part of this project was to try to align the brain activity, so to treat the data and to align the brain activity that you see. So here you've got calcium imaging for a given time. And it has to be aligned with the different markers to try to project this video that we see on a template of the brain region that we know we got from the Allen Institute. And so with this, we are able to understand which part of the brain has which activity at each stage. And so this calcium activity was recorded with quite high resolution, like 600 by 500 pixels, at a frame rate of 15 hertz or 15 frames per second. So that can give us access to activity below 100 millisecond resolution, which is quite fast, much faster than fMRI, which is why I got interested in this study as well. And so what we wanted to do in this study was to test different parcellation, like the anatomical parcellation, and some other one that I can talk about, like the local NMF or ICA, which is just a functional parcellation. Can be used as parcellation. We can do also epoching each time a session that have hundreds of tries and separate the session into training stimulus period or the choice period, depending on what you analyze. And wanted also to test different measures of the calcium activity in order to make the decoding that I'm going to explain later. So the same kind of decoding that we did before with the fMRI, but applied here to see how the calcium activity can help us to discriminate between stimuli or choice, and this to uncover the brain subnetworks that contribute to the processing of stimulus or the generation of the choice response. Right? And so because we had a lot of options in this pipeline, so maybe you don't see that graph so clearly, but that's all sorts of options that we have in our pipeline. And so Damien Jonas did a good job to implement all that in SNAKE, which runs like thousands of jobs in order to make all the possible plots and in a systematic way and minimize the calculation. So we did that in English, and that's something that we are going to make public soon for people who want to use it. And so here you've got example of time series of calcium activity for the anatomical parcellation, meaning that as you see here, all the region that are between the white line, we average their activity for each of them, and we look at them over time. We obtain this kind of traces. So here they are smooth and nice because they correspond to the average of many tries. And you can see the presentation of the 6 stimuli here when you got a raise of activity for some region, and then at this time here, we have peak, that the time of the response. So we first wanted to see whether it could decode using these traces the different stimulus conditions. So you've got left or right, and then either visual stimulation, tactile stimulation, or combination of them. Right. And so what you can see is in the pre-trial period, we've got chance level here, and then as soon as the first stimuli are up, we've got close to 90% decoding accuracy. And this accuracy is slowly getting down during the response period and after in the post-trial. So from this, we can say that we have, there are some patterns of activity in the brain that at each time point vary, and that correspond to a different processing for the different types of stimulation, left, right, or tactile. All right, and so the question was, what is happening in this period and about this decay here. So because we've got a sustained decoding performance here, is it like the same pattern of integration that remained in the brain? And so to do that,
one question, if you go back.
Yeah.
So if you're decoding left versus right, is it like a constantly updated version, or is it like left or right depending on which cuffs come first, or is it just like one label for the entire stimulus period? If you know that at the end you're going to have left.
So here it's so we are using pure trace where you've got all left or all right stimulation. And we are looking at the 6 conditions together, trying to separate them. And that's why the chance level is at 16%, 1/6. And so you're right, because we select the trials that are the pure trials, at the first stimulus presentation, we already know if it's a right trial or a left trial. So there's no accumulation of evidence. Yeah. Thanks for raising the point, because later you're going to mix trials for the trial decode.
But like in the real experiment, it's interleaved whether it's pure left or pure right. So there should be still some ambiguity left for the mouse themselves, right?
Yeah. Yeah, so that's something that we want to check later, is training this decoder on the pure trial, which is left and right, how when you've got the mixed trial, you've got an accumulation of evidence that's going towards saying whether it's left or right. But that's, I'm not going to show that today, but that's where I'm going. Sorry, I just did the simple thing. But then, so if we train the classifier at a given time here on the y-axis and test on another one, so you can see that you've got an area of stability here during the whole stimulation period, meaning that the pattern of activity that correspond to the stimulus, each of these stimulus combination, it is stable for the whole stimulus presentation period for the 6 times. And what is interesting is after this time here, which is after the delay, you've got a reconfiguration, meaning that there's still information to know which kind of stimulus have been presented, but it's transformed. It's another representation in the activity pattern, meaning that it's processing that supposedly concern different subnetworks in the brain. And so to understand first which regions are effective, so we are, and it's a work in progress now, we are analyzing this kind of maps that show which regions contribute to the decoding in the stimulus period. So here what we want to understand is exactly the subnetworks that integrate the stimulus for each type of them at the beginning. And then we can use it later to try to decode the mixed trials and things like that. But for example, here for the left tactile, you've got in yellow, so strong activation of the somatosensory cortex in the right hemisphere, which is fine. You've got this with less contrast now for the right tactile and so on. So now, as you can see, you've got a lot of figure there, and what we are trying to do is to have a way to clean a bit this data to identify the regions with like more contrast and to have a more localized subnetworks. I mean, fewer regions, basically. Now, as I said, we can also decode the choice. And so in that case, we are mixing trials where the animal are doing errors also, meaning that you have to incorporate the mixed trials. And so here you've got the same evolution of the accuracy as before. So when training at a given time and testing at the other time, and here you've got chance level at 50%, and you can see that until the end of the stimulation period, you're still at chance level, and then only after you start to build to be able to decode which thing. So the animal is really waiting for the end of the stimulus period to make its decision. And again, as before, we want to identify which subregions are involved. And this is just, so this decoding like a technique that I showed now, they are just used as a first step to check in the data where we've got information in order to later develop the same kind of dynamic network that I showed with fMRI that you're going to apply to understand how tactile and visual areas, which are sensory areas, they are connected to the secondary motor area here to uncover, to reveal the hierarchy of region that goes from sensory to motor area, to executive area. And so for that, we also want to incorporate some anatomical connectivity that we also can get from the LA Institute. So in order to tune this model, we need to use, like, if you use the same kind of models before, we need to calculate the covariances or the correlation that I said, and the one with lag, without lag, to do the optimization of the model. And so as a current step now, what we are doing is we consider like a single trial activity. And we look, for example, during the stimulus period of what is the mean activity that you can put in a vector and how this can be used for the decoding. And we compare it to like the correlation of this activity during the same period as a marker. And when we do that, we want to understand how specific these functional connectivity matrices are across the different regions. And if they are specific, that means that we can apply our model and tune it using this functional connectivity to capture different communication patterns across these conditions. So that's like a prerequisite step, like a data-driven step to ensure that we will be successful in training the model. And so that's where we are now. So if we look at the decoding between the 6 stimulus condition, with the raw time series, we've got this performance. So for different classifier, but that's not important. So just look at the green here. The mean activity at this performance. And when we look at the functional connectivity, we have here. So compared to the chance level, that's the dashed line, we have like the array above chance, meaning this functional connectivity, so this second order statistic pattern, they are informative. And so that's a good sign for the possibility to apply the model on that later.
But if the MLR is the linear regression,
yeah,
so it's pretty, and you can decompose it linearly as well. Yeah,
but you take the matrix of functional connectivity, then you vectorize it, and you push it into the linear decoder. So that this is just like a data-driven step to see whether there is information there, whether these patterns of functional connectivity that differ significantly across the different conditions, meaning that when you are going to tune the model at the single trial level using these functional connectivity matrices, the estimated connectivity, effective connectivity, is going to capture this difference. So that's really the point of this this step here. All right. So I hope you enjoyed this tour on the whole brain effective connectivity tour. And well, that's as I try to show. It's a good concept to try to bridge structure and function, as well as get markers of brain dynamics that you can also interpret. And so right now, the idea is to try different types of dynamic models. So I've been relying on this Einstein impact process so far, but we now want to try different types of model, because if you want to go for MEG or EG, for example, that have a lot more richness in the signal with different rhythms and things like that, this model is limited. So you just want to expand the diversity of dynamic model that you could use with this technique in order to go towards more richness of applications. Not only fMRI, so calcium imaging is in test, let's say, and then want to go also with the MEG and IAG. And with that, thank you for your attention.